Hello, everyone, and welcome to today's webinar, Next Step in Automating Clinical Genomics, Addressing Liquid Handling Bottlenecks in Cancer Panels, Rare Diseases, and NIPT. I'm Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Automata, the complete lab workflow automation platform consisting of workflow scheduling software with a robotically enabled lab bench. To learn more, visit www.automata.tech. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Alice Tome-Fernandez, Senior Application Scientist, Genomics with Automata. Alice, you may now begin your presentation.
Hey, thank you for attending this webinar. Today I want to talk about common bottlenecks found in automated clinical genomic workflows and how integrated automation can directly address these challenges by further improving efficiencies and improving turnaround times, reducing staff costs. So before I dive into the workflows and some of the examples, I just want to explain a little bit what I mean by integrated automation and a little bit about Automata. People have varying amount of knowledge about these two things here. And this will give a lot of context moving forward about some of the examples I give. So to talk about integrated automation, I kind of want to start about what I mean about automation without integration. So for most people, automation looks like liquid handlers or other benchtop instrumentation that essentially is going to replace a scientist doing pipetting. And things like liquid handlers have massively increased efficiencies in labs. They've made lots of things possible in labs, and they've increased scientific reproducibility, which ends up giving us much better data. And actually, genomics is an application with a good rate of automation adoption. It's very amenable to automation. People already do things manually in plates, so that makes it really easy to transfer over to automation. But despite this, a lot of automation still requires machine tending. And what I mean by machine tending is having scientists that are there to start up the instruments, press go on the instruments, move between instruments, do plate manipulations such as sealing, centrifugation, in order for processes to be completed on these liquid handlers or other instruments. And this often is done by skilled scientists as well. Automation, there is still a learning curve of automation. You're not taught about it until you enter an industry that uses it. So usually these are skilled scientists that are doing it. And this can lead to something, what I always call like scientist dead time, or like scattering your focus, where you might be working on something in between having to do all these plate manipulations, but you can never fully commit. So this is something that I used to do. I used to tend instruments for really important assays, but at the same time, I'd want to go away and do data analysis or start another experiment or do something that you can't automate. And I would find that I would be almost anticipating the next move. And it's not always regular time intervals. So you would have to work out, when do I need to go back? And then you would have to think about, all right, what can I do in that time? And it just generally makes you less focused as a scientist. So these kind of inefficiencies, they can make it really hard to scale. And in my mind, scaling kind of requires three things. So if you were to keep the similar kind of automation regime, you're going to need more people to scale because things need to be machine tending, because you're going to need more instruments, because you would make the most out of the instruments you have, so they'll be at full capacity. And then to enable you to have more people and more instruments, you usually need more space, unless you're lucky enough to already have a lab that you have not outgrown yet, which is rare. And it's really hard to achieve all three of them things. More instruments, often if you're duplicating what you already have, that can be incredibly expensive. People, they're hard to recruit and retain. Sometimes machine tending actually can decrease morale if you're, not the person who's writing the protocols. And then, yeah, the space is just hard at the moment in science. It's expensive and there's not enough of it. So just say you have all of them things, you're still able to scale. But the problem with scaling with humans is humans are very error prone and they can sometimes make mistakes. And if you're scaling with humans, you're probably going to scale your mistakes too. So moving on into integrated automation. So when we talk about integrated automation, we're actually pretty much just talking about the physical and digital connecting of all of these workflows. And then with that, the ability to schedule these workflows. So it's essentially the concept of connecting existing laboratory processes with software and hardware. And then this scheduling allows the movement within workflows as controlled by a single software. So this means that you don't have to be able to access several different bits of software in order to run an end-to-end process. And what this can directly address bottlenecks and increase efficiencies, especially in things like genomic workflows, which can get incredibly complicated and become multi-day. So by doing this, you can then increase your laboratory throughputs, and you can do this without error. So, you know, you're not going to scale the error in the same way that you would if you use humans, because luckily robots are slightly less error prone. So just to talk a little bit about what I mean by the hardware, so connecting things with hardware. And so this can kind of be done in a few ways, but I think for me, the main two things is this idea of transporting between instruments. So that's taken away part of the machine tending aspect. So having something that will physically move them, and then the integration with instruments. And what I mean by that is, yes, like the picture shows, you know, being able to load up a liquid handler with robotics, but it's also being able to coordinate the digital aspect of it. So yes, you can load up a liquid handler, but then you need the liquid handler to know that it's been loaded up and that it's free to run its protocol as planned. So at Automata, the way we do that is the link bench. And so the link bench refers to this as a base unit. So this is a 2-bench system with a robot arm and rail. And then the link platform itself also includes all of the software that allows third-party instrument integration. So something to note, which sometimes isn't clear to people or people get confused about, is that we do not manufacture any instrumentation outside of this. So we don't make liquid handlers, we don't make plate readers, we don't make any of those things. We only connect third-party instrumentation using our hardware and software. So this is the base unit of our product, and it kind of has 3 main features that we talk about. So the arm and rail. So this allows vertical and horizontal movement, meaning that the whole bench is accessible. And this has grippers on the end of the arm that allow SBS format things to be moved around. And actually these grippers are interchangeable for certain applications, but generally this will be moving consumables such as tips or samples in a plate. So this is kind of your way of standing in for the human in this situation. So it, you know, it does what a human arm can do. It moves between things and it gets things across a distance. And then we have number 2, which is our transport layer. So this is quite a unique part of our bench. And this on it has these black things you might be able to see. I'll show a better graphic of this in a minute. But this is able to move things such as plates across work cells. So the robot arm can put things onto a plate and then they can move, and then they can be received at a different point on the work cell and then be moved into a different bit of instrumentation. And that's really important because that allows us to have lots of different configurations of this bench. So if you have 2 benches back to back, the robot arm can't reach the bench at the back, but the transport layer can. And same for around corners. And I'll be showing some examples where you can see where the power of this comes into play. And then also the third thing to point out, which I always think seems quite obvious at first, but this is the bench top. And the bench top has been very deliberately designed in a way that all of the instrumentation can face towards you. So the robot itself is a co-robot, so you can work alongside the robot and you're in no danger of getting hurt by it. And what this means is that you can also work with instrumentation placed on the bench. So it'll be facing you, so you'll have full access. And that means you won't lose, some of your more complicated, expensive instrumentation to automation. So you'll be able to go up and use it manually as required. As long as it's not being used at that very moment by the work cell, you can go and use it. And that's a really important point. So it means that you don't need to duplicate instruments for manual and automated use. And another design aspect about these benches, which I want to point out, is their size. So they're 1 meter lengthwise each, and that's very similar to standard laboratory benches. So in labs where we have gone in and automated workflows for them, it hasn't required more space because they're able to fit in 1 for 1. So it's a really important part that sets us apart from some integrated automations, that we try to make it so they can fit seamlessly into your lab. And then here's just a quick animation showing that transport layer that I was talking about. So the arm is able to move things down, it's able to move across benches to the other side, and then able to be received by another robot. And what that means is that your robot arms aren't overutilized. It means that they can carry on doing a process at the same time that things are being moved across the work cell. And then I just want to touch a little bit on our software and how you can build workflows on there. So we have a node-based system, which is how you can link all of the instrumentation in whatever combination is required by your workflow. So each of these nodes actually represents an instrument. And then within that node, you can set the parameters. So you can set things such as loops, time constraints, data transfers. And then you can control in which order all of these things happen. And the transport, so using the transport layer or the robot arms, is handled implicitly by the system. So you don't have to sit down and plan every single movement through the work cell. And then what the other thing that the software will be able to do is it will be able to control each of the instruments that you've put on your work cell as well. So you will not have to go in and start the process manually. And another thing I touched on earlier was this idea of scheduling. So this is really important to enable workflows. So once the workflow has been designed, as I just showed in that previous slide, your runs will then be visualized. And what's shown here in blue is just showing your run, what's running. It probably won't be called run, it will probably be called something a lot more useful. But that just shows which run is running, and then you're able to open up, like shown on that previous slide, that previous animation, to look at a more detailed view. So the software is able to kind of map the labware journey through the system based on your workflow and all the parameters that you set before. It'll be able to run multiple batches and different workflows completely simultaneously. So in this situation, like I said, that blue one's just the run and the run name and what's currently running. And then when you open it up, you can see that actually you've got 3 batches going through here, so, and they're coloured. So the lighter green is one batch. You can see something's being washed, sealed, shaked, and then peeled, washed, etc. And then batch 2, once that first watch is finished, the software will be able to say, okay, I now know that that's free for my second batch to begin, and it will schedule it in a way that doesn't stop any of the other time constraints required by the first batch. So you can see it does a wash, knowing full well it will then be able to seal and shake at appropriate time without disrupting the first run or the first batch. And then the same for batch 3. I will touch a little bit more on scheduling and how it works in our system, but that's just to give you an idea of how you can link things whilst maintaining a good level of efficiency. So now I've kind of introduced integrated automation a bit more. I want to talk a little bit about liquid handling bottlenecks. Obviously, this isn't, all of the liquid handling bottlenecks that exist for all workflows. I've probably missed some, or there's some that don't apply to every workflow, but these are the ones that we find ourselves talking about most with clients. So I've got 3 up on the screen here. So the 3 I want to address is the inefficient use of complex liquid handlers, the disconnected workflows. So the way that we often find ourselves splitting workflows across different devices or liquid handlers, and then also just doing single plates, so single plate batches, so only processing one plate at a time. Essentially, this is usually liquid handling capacity determining your throughput. So the first one I want to focus on is this inefficient utilization of liquid handlers for throughput. And what I mean by inefficient use is, in this context especially, is kind of putting things on deck that don't need to be on deck if you could link things to things off deck. That's a very complicated way of saying it. And this is going to be mainly in the context of trying to get things through your liquid handler as quickly as possible. So this is going to be focusing on throughput, because obviously, you know, having things on deck might work if you don't need to get through more than one plate a day. But we find that a lot of our customers and the people that we work with, actually, you know, one plate a day isn't enough for them anymore. And actually, throughput is an important consideration for clinical genomics. You know, as increased throughput often positively correlates to reducing a turnaround time, or, you know, reducing staff costs. And it's often the thing that is stopping labs be more efficient. So coming back to what I mean by inefficient utilization. So it's using the deck for on-deck devices that aren't required to be on deck. So, you know, sometimes if you weren't working with a liquid handler and it wasn't a liquid handler-centric system, you probably wouldn't do it on deck. So some examples that I've put here is an on-deck devices. So this one's an on-deck thermal cycler. Here's an on-deck peeler. And these take up quite a lot of space on the deck. You know, this takes the place of almost 3 SBS format lengthways, maybe 1 or 2 sideways. And then you have the control box as well, which also takes up a lot of space. And that means you probably have to get a bigger liquid handler and able to do that with your current workflow, or you have to take some other things off deck, or you have to do more deck setups. So the system that I'm going to use to show some concept over the next few slides is an NGS system. This has been designed to run things such as panels. So in this case, cancer panels, but it could be methylation panels or something like that, or rare disease panels. And what I want to do is kind of show how off-deck devices have been used to increase throughputs and efficiencies on the system. So what you're looking at here is actually the pre-PCR work cell. And on there you have a reformatter, which is essentially just a smaller liquid handler that's capable of handling tubes, a larger 96 head liquid handler, a peeler, a sealer, a thermocycler, a centrifuge, and a cooled plate hotel. And so you'll see here 2 instruments are highlighted, and that's to show the things that we have added on deck to make the liquid handler work more efficiently. So we've added the reformatter, and this is responsible for reformatting reagents from tubes to plates, or samples from tubes to plates, so that the larger liquid handler is able to spend more time just doing large liquid handler jobs. So that might be using its 96 head to stamp, or it might be to do complicated things such as bead cleanups. So by having the reformatter put things into the right format and then be moved onto the larger liquid handler, you're freeing up the space on the liquid handler. And it also means multiple plates can move through the system. So the second thing I want to show, also on the pre-PCR system, as you can see now highlighted as the 96 head liquid handler and the thermal cycler. And by moving the thermal cycler off deck, things like long or overnight hybridizations will not block off the instrument to begin or finish a separate process. And because it's an automated instrument, there's no manual plate movement to it or manual program selection to kick off the hybridization. And the other really cool thing about using something like this is actually you could add a 2nd, 3rd, or 4th thermal cycler off deck, and that doesn't negatively impact your deck space. So what that means is you could add a shelf in there and stick one above, and that instantly means you're able to get 2 plates up to the point of a overnight hybridization without having to rely on getting another liquid handler. And that becomes really important for super high throughput applications and when you can't get more deck space or you can't get a second liquid handler. So now we're moving on to the post-PCR system, which actually looks very similarly to the pre-PCR system. So we've got this L format. This is actually a really good use of the transport layer to get around corners. And we've got a lot of similar instruments, except from you might notice that the reformatter has been replaced with some QC instruments. So a plate reader to quantify your final libraries, and then also a fragment sizer to check that we've got the right size for sequencing. And the reason I wanted to show this is what it allows. Again, if you were to integrate a plate reader on deck, that's going to take up quite a lot of space. It's going to be a complicated integration, but also what it means is you're more likely to only be able to integrate one type of QC. So in this case, you might just be able to get a plate reader integration on deck. And the problem with that is that might not give you enough information to effectively pool your libraries. So if you do have to add another QC metric on there, such as fragment size, then you're going to have to either create a manual process out of that. So you're going to have to have manual plate movement, or you're going to have to, well, not do it essentially, unless you're willing to get a second liquid handler with an integration on it. So secondly, on the post-PCR system, I wanted to highlight the incubator and how that's used with the sealer and liquid handler. So by including the incubator, we're able to store plates at safe stopping points. So this is really important because it means there's less need for cooled positions on deck. It's always good to have a cooled position on a deck designed for this kind of workflow, but it does also mean that other processes can't use it if it has to stay there overnight after a process, or it has to stay there while you wait for a manual interaction. And it also means that things like library pools can be immediately moved and called off deck with or without cooled positions. We know that's important because of things like index hopping, and once you get to the end of a library prep, you know that you've spent a lot of money and time on getting that library pool to where it is, so you don't want to have to risk leaving things open and on deck. So using a sealer and putting it into an incubator puts it into a really safe place where a human can then actually interact with it to continue the process. Another bottleneck we can address is how the commitment to a liquid handler can be a commitment to disconnected workflows, I guess. If there's only one liquid handler, you will need to do multiple deck setups. If there are multiple liquid handlers, you'll likely need to move between them. This is done manually, and it can be backwards and forwards a lot. And it can create a bottleneck as it's dependent on humans not hesitating. Sometimes these need to be done quickly. They need to be planned, which is actually really hard to do manually. And, you know, all of these hesitations, different speeds of movements and competencies can actually end up making things less reproducible, and just generally all of these inconsistencies can add up. So the way I want to talk about this is actually using quite a large end-to-end process. So that's NIPT, so non-invasive prenatal testing. So what this is I won't go into too much detail because I imagine a lot of people here will know about it. But the workflow is at the top. But essentially, you will take a pregnant person's DNA. You will isolate the plasma from it with the intention of getting a high amount of fetal DNA from the blood. So pregnant women will have a fraction of their blood will contain the fetus DNA. So you need to isolate that from the plasma. That's done by spinning down whole blood. And then the cell-free DNA needs to be extracted, done similar way to normal DNA extraction. And then you want to find out how much of that DNA you've got. You want to normalize it. And then that will go into a library prep process like the one that we saw before. So that's library prep, QCing the library to make sure that you have enough and of good quality, and then pooling that library for sequencing. We haven't included the actual sequencing in this because that is not an automatable process at this time. It's also a very long process. So you cannot always increase the throughput of sequencing with just automation alone. And the thing about this process, as you can see, it's quite a long process, and that makes it harder to scale. You know, the longer the process is, the harder it is to do a lot of it. I think that's fairly straightforward. But actually, there's already quite a lot of laboratories that have implemented liquid handling solutions to increase their throughput, deal with increasing demand for sample numbers. But there's still a lot of disconnect between all of these vital processes. So what we've tried to depict here is that, okay, so just say this is done in 3 different rooms. So you might have your plasma isolation done in one room where you'll receive your samples. You'll have to spin down all of the whole blood. If that's, you know, 80 to 88 samples, that's 88 times that you're going to have to get things spun down. They don't have an 88 capacity, usually these centrifuges, so you're going to have to do multiples of them. That's going to require someone. And then if it does go onto a liquid handler, you're still going to have to load that liquid handler. And then as you start having to do QCs, that's also going to be a manual movement. And then you might have another room for extraction where you're going to, in a good situation, you'll have a benchtop extractor, but then you might still need a liquid handler to set up QCs. You might have to go back to a room to use their QC equipment. And then you might have a third room. And then in that third room, that might be where you're doing a lot of your library prep process. So a lot of it may be happening on a liquid handler, but then you're requiring PCR machines and QC instrumentation. And often there's overlap. So you might have 3 people doing this process, but you might need 2 here because of the sample integrity situation. So it might be that you need someone to make sure that things are being moved to the right tubes for reformatting. And you might have to go back and forth between rooms. And this can lead to bad traceability, which is obviously for things like NIPT, it's really important that you know that your samples are safe. So one way we see integrated automation tackling this is actually moving them into work cells. So what that means is that you don't have that constant movement between instruments. You can control the flow of movement. So in this example, we have this first work cell, which has a large liquid handler that's capable of doing the plasma isolation. But we also have an automated centrifuge. So what that means is that you're able to load your blood samples into here, but then actually the centrifugation can be automated. And what this also means is obviously it takes away touch points. It means that you only need 1 person there, because actually you can ensure traceability because it's a robot doing it. The only reason you usually need 2 people for handling of clinical samples is in case somebody makes a mistake. But the good thing about programming a robot is it's always going to put the right thing in the right place. There's no hesitation. So in this first thing, you'll be isolating the plasma from blood and reformatting it into a plate. And then that would be ready to move on to the second work cell. And then in this work cell, we're able to use off-deck extractors in order to extract the cfDNA from the samples. And this is a complete walk-away process. So we have the extractors here. We have the robot arm that will do all of the tending, swapping between all of the reagents. And then we also have a dispenser that will be able to fill up all of the plates with the reagents that are required for the extraction. And then also on the same system, we've included its own little QC station with a qPCR and liquid handler. Advantage of this is that as soon as a plate's ready, it can then be QC'd, and then that data can be automatically uploaded, and a work list can be created in order to do the normalization. So you're actually doing quite a lot in one work cell. And remembering this is only 3 benches long as well. So it's not a large work cell, and actually you can kind of see how it could fit into an existing lab. And then in the last one, we have the library prep solution. So with a liquid handler, the smaller liquid handler, sealer and peeler to do all of those mid-workflow steps where you need to seal peel plates, and then maybe you need to spin it down. There's a lot of them in most NGS workflows. We have an off-deck thermocycler, and then we have that safe storage point, so a temperature-controlled incubator. And the good thing about doing it in this way is that you can use the best instrumentation that's required for the job. So you're not stuck using one that's supported by the liquid handler. You're not stuck using one that can fit on the liquid handler. Often when you have integrations on liquid handlers, they tend to be lower capacity. So you might not always be able to do 96 samples at once. But you're able to do it in a way that doesn't take up a lot of space in your lab. And then the other advantage of this is actually you can build up. So you don't necessarily have to get all three of these at once. You can get one and then you could get the other. You could get part of a work cell and then add on another part. So for this NAE one, you can see we have the extraction part here, but then you could actually add the QC part later if you start realizing that actually your bottleneck now becomes the QC of all of these. And then the next step in this process might be joining them all together. So I just showed 3 separate ones. Why would you join them all together? Actually, if you were to combine all 3 of them, you actually would take up more space than this one, because you can start to get rid of duplicate instruments. So you won't need to get 3 separate peelers. You can just get 1 peeler. You won't need to get 3 separate centrifuges. You can just get 1. And because all of the scheduling is going to be handled by the software, it means that actually you don't need duplicate instruments, because it's not going to start a process until it knows that it's going to be able to do the centrifugation when it needs to be done. And then you end up with a true end-to-end solution when you do it this way. You put in your blood at the beginning and you end up with a library pool ready for sequencer loading. And what this means is that you only have one setup, and one setup with 1 staff member required. So one setup point of which would be your manual interaction, and then all DEX setups can be handled by the system. So that's why we have a lot of these incubators and this storage. So this is like a plate hotel that's able of holding consumables at room temperature. So that can be used to unload tips, and then they can be moved across the system using the transport layer to underneath your liquid handler, and then the robot arm will be able to start loading up your liquid handler. So you don't need that continuous DEX setup. And it means that all plate movements will be triggered when the process is that they're being used on are completed. So a lot more simple way of saying that would be, once your reformatter has reformatted, then the robot arm will know because of the software, and it'll be able to move it on to the next process. So the final thing I want to talk about is the single plate batch. So this can often happen with liquid handlers. They have the capacity of doing 1 plate at a time. I will say, like, this isn't true always. You can design them in the way that they can do more than 1 plate. That obviously becomes more of a software problem, which is often not able to be handled by the customer. Requires apps time. And also a lot of these workflows are originally designed to contain all of the reagents and consumables, which can be fit on the deck. So what I'm saying by that is, if you are going to start doing 2 plates, you're probably going to need double the tips. And actually that will then require more deck setup. So it becomes a lot more hands-on, because the deck is your determining factor for throughput often. And then if, just say, you are able to get more plates on a liquid handler without any other automation, you would then more likely need to start buying more off-deck instrumentation. So if you are doing things off-deck, you might need to start getting more thermal cyclers and things like that. But essentially, if you are considering a workflow where it's a 1-plate workflow, if you then want to turn it into 2-plate workflow, and you haven't got the deck space, or the software isn't capable of doing that, you're probably going to have to get a second liquid handler, which, like I spoke about before, may require another person, and it will certainly require more space. And it's a large capital expenditure as well. So here I'm going to talk about the NIPT example again, while it's still fresh in all our minds. And obviously it's a long multi-step process, has time constraints, and a real big need for precision. So what this graphic is doing is showing the differences between a single liquid handler run and using an Automata work cell. So in the single liquid handler line. So it's all being shown to happen on a single liquid handler. So this means a maximum of 1 plate can be processed in 8 hours. So it's actually pretty cool, right? You can do blood fractionation or plasma isolation all the way to library pooling on a single liquid handler in 8 hours. That's not bad going. But what it does mean is if you want to increase that throughput, you're going to have to get another liquid handler because it's being fully utilized. Or you're going to have to wait until the next day. And then that puts quite a solid ceiling on what you can do with that one liquid handler. And because so much is happening on one liquid handler, that also means that you're going to have to pause and do multiple deck setups as well. There's no way you'll be able to keep all of your decks for the whole process on a single liquid handler, especially not for a genomics workflow. You're going to have to keep on replenishing them filtered tips, right? But in the integrated automation workflow, you can kind of start to see how the batches or plates can be staggered and the processes can start to run in parallel. And this allows that throughput increases. So by spreading the batches across multiple different instruments, this allows plates to enter the workflow at different stages without the need for duplicate instruments. So just say here, my blood is getting fractionated, and then it's moving over to start being extracted. The system will then know, okay, right, great, I know I've got to do a second plate. So now I know that the path is clear for me to start this process. Just say this takes an hour, this takes an hour, and then this knows, okay, if I start now, by the time I'm done, so by the time the blood is fractionated, I know that then the extraction part of this workflow is going to be free for me to put on the next plate. And that means you can start to get more batches out in the same amount of time. And then you'll see another thing about this is that actually we have a batch going overnight. And that's a really unique thing about our system, is that actually you can start to run things overnight because you don't need the human there doing the plate manipulations. So that means that you can have a batch that goes over them 8 hours. It makes no difference to the system. It doesn't care that it's nighttime. So all of these automated plate movements means that these can continuously be linked. And then this can, in theory, go on for a long time, right? The system's not going to get tired. It's going to start planning for as long as you let it. And because we're putting them large plate hotels and cooled plate storage, it means that as long as there's enough loaded in there, then the system can keep going. And one of the main things that allows all of this to happening is coming back to that thing I was speaking about earlier, scheduling. So this is just an example of a workflow, and you can see that it's kind of showing that, you know, your plate might start here, but then it's going to have to go to 1, and then it's going to have to go to this liquid handler on 2, and then it's going to have to go to 3, but then it's going to have to go back here, and back to this plate washer, and then so on and so forth. And actually, if you do that as a human, it's complicated. You know, that's you walking across a lab and ticking things off on a sheet, and, you know, maybe noting the time of something happening. And that's something that our software can take control of. So in a nutshell, scheduling is the algorithm that controls how different pieces of labware, such as a plate, moves through a workflow over time in a way that meets all of the parameters that I talked about earlier and time constraints. And this will allow the parallel running of the same or sometimes different workflows on the same work cell. And essentially, it's going to do all the coordination. It's doing all the coordination that sometimes we do when we're multitasking, but we don't even think about it. And it's able to call these instruments, get things to these instruments, but also tell the instruments to start working, right? Or take some data, or stop working, or get ready for another plate. And it's actually a really complicated thing to do. So our scheduling algorithm is actually what we're kind of calling the next generation of scheduling algorithms. So schedulers aren't a new concept. They've been essential for integrated automation since labs begin to integrate anything. And they're typically done one of two ways. So they can be a static scheduler, which is where they will plan the run ahead of time. So you would get a Gantt chart, but This is fine if everything goes to plan and everything takes the right amount of time. But what this means is unable to deal with change. So if there's an error or you need to change parameter, it's going to have to recompute, and that can take a long time and can cause essentially a process to not be viable anymore. Or some schedulers are more dynamic, where there is no planning ahead and it reacts to real-time events, but then that means it can't plan to include all of them parameters I keep talking about, so such as time constraints. And both ways have their pros and cons within the scheduling world. You know, it might mean that you actually have to use 2 different schedulers for 2 different processes because time constraints are more important in one, or speed is more important than the other. But what we've done at Automata is actually try to directly address this issue by combining both approaches. So what our scheduler does is it does that planning ahead using solvers that are embedded in the Link platform. And these are modern fast solvers, meaning they can reschedule very quickly, like in seconds. So it's constantly looking ahead and planning. But then it's also combined with an executor, which is something that is able to continuously check the work cell to make sure that, okay, this bit of equipment is available and this process will take this much time, and actually I can do the schedule that I've proposed, or I can't do the schedule. And you might be wondering why I've put a map on here. And I think that's to show like what the result of this new type of scheduling is. So it works very similarly to Google Maps. So it will plan your route and you'll be able to see the plan, just like when you get in your car and say you're going to go somewhere. And if you say, I really don't want to use toll roads, it will plan for you to not use toll roads. But if you also then take a wrong turn, it is actually able to recalculate fast enough that you're not going to arrive at your journey, arrive at your destination, you know, an hour later because it couldn't do it in time. It's going to give you new instructions straight away. So you're not going to, I don't know, miss a flight because of it. And that's what's been shown here. So you can see here, this is the scheduler, similar to what the pictures I showed before. What it's able to do is it's like, oh, something's happened here with this instrument. I better just shift everything, but still make sure that this time constraint is kept. So it doesn't just go ahead after the change and then have to delay a time constraint. It keeps that time constraint. And that's what allows us to do these complicated, you know, multi-system, sometimes even multi-day workflows. So I just want to finish by talking a little bit about an actual customer workflow, and how a few of these concepts were brought together and able to give them the metrics that they need, and the improvements in their workflow that they desired. So this is a system that is able to do end-to-end cancer screening, essentially. It can do multiple extractions and library prep workflows, and result in a sequencing pool ready to go on a sequencer. And this is the pre-PCR part of that system. So it combines extraction, the QC of that extraction, and the pre-PCR library prep. And you can see the system has several larger liquid handlers on there for some more complex tasks. So, you know, for all of that plate stamping, for some spry cleanups and things like that. But we've also put a reformatter here, and this is able to take some tubes and reformat them into ways that then they can just be moved straight onto the larger liquid handlers. They can be stamped, and they can be processed, and then the workflows can converge as needed. And you also see down here we have 2 extractors below one of the liquid handlers, and these are off deck. And this means that actually the extraction can run in parallel to other processes. So, you know, it can be, you can start extracting something whilst you're still reformatting some patient samples, for example. So what that means is that you're not going to negatively impact the throughput by doing more on the system. And then this is the post-PCR of the same system. And this is able to finish off the library prep for all of the workflows on there, and create the final library pools. So what I've highlighted here is the use of smaller liquid handlers. So these can actually be scheduled to allow multiple workflows to be run on the system whilst sharing some of the off-deck devices. So you can see here there's like a little pool of cool plates. All of these can share them. And what that means, you can have multiple processes and they can use all of these off-deck devices without negatively impacting the throughput of one of the others. And then also I've just highlighted here, we have multiple thermal cyclers down here as well. That's just an example of using some off-deck devices in order to make the most out of the liquid handling capabilities here. And you can see on here, you've got 3 liquid handlers on a relatively small footprint. You wouldn't be able to do this with some of the liquid handlers that might be required that have cool plates and thermocyclers and sealers and peelers and centrifuges on, right? You would probably get 1 or 2 on that in that same space, probably more likely 1. And then bringing that all together, this leads to these metrics essentially. So using these concepts of integrated automation, this system was able to get to these key metrics. And these weren't being hit before, right? They had a lot of these existing liquid handlers and a very liquid handling centric automation, but they weren't getting this throughput. So what they were able to do is they were able to triple the throughput, which allowed more clinical screening. They were able to reduce the cost reduction per test because of the reduced staff cost as they scaled. And then the staff quality of life was also improved, right? There was a lot less manual interactions, and that led to less human error. Thank you so much for listening to this webinar, and I look forward to hearing some questions.
Thank you, Alice, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for today. Let's go ahead and get started. Our first question asks, can you integrate shorter workflows?
So yeah, so obviously the ones that were shown on my slide deck there, they tend to be the longer ones, so all of the clinical genomic ones, so multi-day ones. So I guess something that might be considered a shorter workflow might be just taking a portion of that out, so maybe just the QC workflow or something like that. And actually that's something we quite regularly automate at Automata. So that would probably only be like a 1 or 2 bench system with liquid handler, plate reader, peeler, sealer, and we're always really happy to do them. They're often the start of creating a more automated lab. So yeah, definitely can.
Wonderful, thank you. Our next question here that we have asks, has integrated automation already been used in healthcare settings?
Yeah, so I think yeah, so it has, yeah. And I think sometimes people worry a bit about that in healthcare settings because obviously it's a really complicated thing, integrated automation, and people worry about being able to like validate their clinical workflows on there. But yeah, we ourselves have got a system deployed in the Royal Marsden Hospital in the UK. There's quite a lot of information about that on our website and things. So yeah, we can do that and it has been done. And you know, we've got the experience in validating these workflows in a clinical setting as well, and it's been really valuable for us to do that. So now when we're approached by other clinical clients, we have kind of some tools in our back pockets.
Wonderful. Thank you so much for that. It looks like we have time today for just one more question, and that question asks, what if I want to change a workflow once the system is in place?
Yeah, so there's a few ways we can approach that. If you've kind of thought about it, so like for the example I gave earlier, where you might start with a smaller work cell for something like QC, and you might be thinking, I want to start with this smaller work cell, but I know I'm going to want to build it up. At that point, you might have left some gaps, or you know, left a space to add more benches. So in that case, we can just add the instruments onto them gaps you've left. Otherwise, even if you haven't done that, it might be the case that actually you can just add another bench onto your system or several more benches onto the system to make more space for new instruments. And then you can build up and change your workflow that way. Or alternatively, if you have a workflow, but you want to actually change that exact workflow rather than add to it, we can actually swap out your instruments as well. So how that would work is that we get your new instruments and then we can physically swap them out. One of our engineers can help with that. But actually, when it comes to changing the workflow, the customer will be able to do that. So like I showed earlier, that node-based system, you would be able to build that new workflow out using the newly integrated instruments that way. So yeah, it's pretty easy for us to do, and it's something we can do well and quickly.
Great. Thank you again, Alice, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Automata, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions that we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.